everybody, welcome to the third vodcast in our energy domain. This one's a biggie, gang, so, strap in and hold on to your seats because we got a lot to cover in this one, because here is where we are going to introduce the idea of photoautotrophic energy processing, otherwise known as organisms that utilize sunlight and convert it into chemical energy, aka photosynthesis. So let's get rolling because we got a lot to do here. All right, so here's the best big question. How do we process energy? It starts here. Boom, done. Quick recap, all right? We know about the needs that energy and organisms have. So this should be kind of a reminder of all of this. So reciprocal relationship between chemioheterotrophic nutrition, photoautotropic nutrition. If you're unsure of those terms, you best look them up because we're going to be using them a lot, okay? So, the inputs of one are the outputs of the other. So this is part of that exchange, that input-output of energy. And so this is what accounts for this really interesting dynamic of photosynthesis and cellular respiration, the fact that they are opposite of one another. So light makes us food. So without light, we wouldn't be able to eat. And I know you want to eat because you're doing it all the time. Okay, so here's that idea of that input versus output. Okay, light comes in, chloroplasts use it, convert it to chemical energy, mitochondria then break that down, release the energy, build up ATP, we release heat, we send water and carbon dioxide back into the atmosphere, which is then used again by the chloroplasts, and the chloroplasts release oxygen, which is used by the mitochondria, and it's just a cycle. So we know this. Here is the fact. Photosynthesis goes one way, respiration goes the other. And we're going to explore what the heck is going on with this big arrow, because this arrow, in both processes, means a lot. There is a lot going on in these arrows, and you're going to have to be able to know what it means. So plants aren't the only photoautotrophs. Remember that. I mean, up until this point, we've really focused on plants. Why? Because we look out our window and that's what we see. But they are not it. There are lots of other things. Okay, in fact, plants aren't even the main oxygen producer for our planet. Most of it is generated by cyanobacteria and aquatic protists that are photosynthetic as well. So here are a couple of examples that we have here. So we have sulfur bacteria, cyanobacteria, unicellular algae. Algae is such an interesting organism. And then you have seaweed. All of these are responsible for, blasting our planet with oxygen. However, plant anatomy, yeah, we're used to it. So photosynthesis happens at the leaves. I know, you're all going, durr, but it does. So organs that are specialized for this process, that's their job. So the mesophyll cells are full of chloroplasts. That's where it happens. So we're not only talking about a cell being responsible, it's a structure inside the cell that's going to make this happen, which you guys know because you know your organelles at this point. I don't need to tell you that. Light is key in this. That's why it's called photosynthesis. So photo means light. Light is energy. But not every aspect of the light energy that comes in is something that's usable. So let's talk about the type of light that we do use. And the visible light spectrum is what our chloroplasts are going to use. There's lots of radiation that comes in with light. It's particle and wave. And it is produced by the movement of electrons. As electrons jump orbitals, boom, light. So it's just that visible piece is a very, very, very small piece of a very, very big pie. So the chlorophyll, that is the pigment that is housed within the chloroplast. It's a pigment, which means that it is a protein. Okay, so any molecule that interacts with light to produce a color is known as a pigment. We have two types, chlorophyll A, chlorophyll B. It's not the only pigment found in chloroplasts, but it's kind of the main one. You have accessory pigments that also are there too, carotenoids, xanthophylls. These can help to absorb wider ranges of visible light spectrum, and it helps to protect the chloroplast from damaging light as well. So we're trying to kind of keep things happy. So why are plants green? Sunlight comes in, the chlorophyll and the chloroplasts are going to absorb pretty much all the waves of light except, you guessed it, green. Okay, so they're going to bounce that back. So in terms of what they're capable of taking in, they're going to suck up green light or excuse me, they're going to suck up every light but green, yeah, and they're going to bounce and throw the green light back to our eye. So they definitely like colors within the reds, the blues, the purples, and you can see that there is really, really, really a low photosynthetic activity that happens under green light. Okay, you also have Engelmanns Experiment, and Engelmanns Experiment took algae, placed it in varying, these cells of algae in varying light frequencies, and allowed aerobic bacteria to hang out. And what they found was you got huge amounts of aerobic bacteria growing in different areas. You can see huge amounts over here and huge amounts closer to the 600-700 side. And that's because what is a waste product of photosynthesis but oxygen? And if I'm an aerobic bacterium, I am going to want oxygen because I'm going to want oxygen to be able to help with my metabolism. So the more oxygen that was being produced equaled the more photosynthesis that was happened, equaled more bacteria there. Notice very, very lacking amount of bacteria in this center section in the green area. So that's Engelmann's experiment. So let's hit the road to the light. Photosynthesis, a huge process of varying degrees of things that are going on here. So it's going to be really, really key for you to get the overall big picture, to know what happens during the light reactions, during what happens in the light independent reactions, and to really have an understanding of what each part is used for, as far as memorizing specific aspects and reactions that are going on, but you need to be able to analyze them. So, Groovin. Let's do it. It is an anabolic, it is endergonic. So we are going to take carbon dioxide, we are going to reduce it. If you forgot that, Leo says GR, right? We got to remember our reduction oxidation reactions. Carbon dioxide is reduced into glucose, where you get water being oxidized. So the reducing agent is water, the oxidizing agent is the carbon. Okay, so just to kind of remember that. So, are you ready? Are you sitting down? Do you have your notebook out? Good. Have you turned me off yet? I'm just kidding. All right, I know you guys can do this, and some of you may remember some of this stuff, and for some of you guys, this is going to be new. So that's why this is here. Come back to it as often as you need to, and you have the text as well. All right, so again, our chloroplasts structurally are a series of membranous discs called thylakoids arranged in stacks known as grana. The grana are inside. The key to a chloroplast working properly is the immense number of folds. What does that do? Increases surface area, gives me lots of membrane folds to be throwing things through those membranes. We have a fluid-filled area that's called the stroma. And this, we have photosynthetic prokaryotes that are going to use special membrane regions to accomplish this because, we know the theory is that chloroplasts at one point in time probably were their own bacterial cell. So this is why this is going to happen the way it does. Yeah. Here's the big picture. It's a two-part process. You got the light reactions, and then you have the Calvin cycle, aka the light independent reactions. So that's kind of how I'll be referring to them. The light reactions only are occurring in the thylakoid membrane. The light reaction, the big part of the light reaction is really to grab and harness the light in order to blast apart a water molecule. And that water is going to then, when you blast apart the water molecule, that is going to supply a needed amount of electrons to create an electron gradient, electron transport chain that will help drive the production of ATP molecules as well as NADPH, which is a really, really important cofactor. So NADPH is a reduced molecule from NADP plus that is reduced through the gaining of the electrons released from the breaking down of water molecules, and then is used as coenzymes necessary in the Calvin cycle. And in the Calvin cycle, which occurs in the stroma, in the space, the ATP and NADPH that's produced in the light cycles are then used to help put together sugar molecules. So there's that in a nutshell. Shall we get specific? So let's get specific to the light reactions. So light in chlorophyll is going to give us the electrons. The electrons are super necessary for this process, setting up an electrochemical gradient that's going to help bounce these electrons through pigment, and that's what they do. They bounce through the pigments and through the membrane in order to do their thing. Okay, so when photons of light come and react with chlorophyll, you can have electrons in magnesium, you can actually excite them. So this happens with about 1% of all the sunlight that strikes the surface of the earth, comes in as photons, and an isolated chlorophyll will fluoresce, glow, when exposed to light as the excited electrons return to their ground state. So you get this excited state of electrons releasing energy, releasing the photon, and that is what is going to sort of chain react through the molecule itself. So with photosynthesis, we have huge amounts of membrane proteins that are necessary for this embedded, which is what we call photosystems. There are two of them, photosystem 2, photosystem 1, that run within the membranes of these thylakoids. So hugely complex series of proteins and pigments that are embedded in this membrane. The incoming photons hit what's known as a reaction center. The chlorophyll molecules then produce excited electrons, which are transferred through an electron transport chain, if you remember that. And this then sets up the electrons to be able then to be captured and to help reduce NADP plus into NADPH. So what goes in, what comes out? Photon, light comes in, okay, we're going to excite these, okay, we're going to get basically exciting in the reaction center complex that's going to help to bounce the electrons through them and allow for that harvesting of the light to basically blast apart that water molecule. So let's look at our two series of photosystems. Photosystem 2, central chlorophyll, works best at light wavelengths of about 680. It's weird to call it photosystem 2. It's the first in the series. So photosystem 2 works at the start of the electron transport chain, where photosystem 1 is at the end. And chlorophyll is not going to have electrons return to it because the electrons are used in reducing NADP+. So since that's the case, constantly new electrons are needed. So the water is what provides us with our source of electrons. This is photolysis. Light comes in, boom, blast it. Blast the water molecule apart, creating 4 protons, 1 molecule of oxygen. That is what gets spit out first. So the oxygen waste product is released first in photosynthesis. Conversely, in cellular respiration, oxygen is used at the end. Again, that flip, all right? And so for every 2 water molecules that are consumed, that's what we get. And we get, those electrons to be able to bing their way through, okay? So oxygen gas is released, major import for aerobic cellular respiration. So everybody take a deep breath. We're grooving. So chemiosmosis, this is what's going to occur once we have the electrons moving through the transport chain. They're going to provide the energy for the chemiosmosis, almost identical in cellular respiration. Protons get pumped through into the thylakoid space. We're going to use facilitated diffusion of the protons back, again, aka protons, hydrogen ions. Keep that in mind. Every time you hear proton, hydrogen ion. So ion needs facilitated diffusion. Okay, back through, and setting up that gradient of protons that run through those proteins are going to help promote the synthesis of ATP. So kind of on a notable difference here in terms of ATP production in a chloroplast versus mitochondria. In respiration, the energy comes from the oxidation of glucose. In photosynthesis, it comes from photophosphorylation. So the energy is coming from the light in the case of in a chloroplast, it's coming from glucose in mitochondria. So just a little aside thing. So here it is. Pause me for a second. take a look at this, do a little bit of drawing. You know, we've got the light coming in, we bust apart the water molecule, we set up a hydrogen gradient. That hydrogen gradient is what then flows through ATP synthase to promote the formation of ATP. The electrons that are released run through the both photosystems, then going through NADP reductase, which is the enzyme that is going to reduce NADP plus into NADPH, NADPH and the ATP then head Calvin cycle. So that's just a little bit of some difference in terms of what's going on mitochondrially versus chloroplast. So just take a peek. Flow of electrons, this is a non-cyclic flow of electrons. They start in one place, they end in another. There is no return back. This is why water is so important, because water is going to provide us with our constant supply of those electrons as we go through. So they move from the photosystem 2 through 1 via the electron transport chain. They then get transferred NADP plus through the reductase, that enzyme. They're going to reduce it into NADPH. This is going to produce, at the end of the day, both ATP and both NADPH. Key components for Calvin cycle. There is also a cyclic electron flow that happens here, just because why not? Confuse you a little bit. Electrons move from photosystem 1 to the ETC before they return back to the photosystem 1. In this case, we are only producing ATP as a result. This part doesn't require water, because we can use and reuse the electrons, right? Again, what's the purpose of the water? To supply us with the electrons that are necessary. So Calvin cycle requires 9 ATP molecules, 6 NADPHs for every single sugar molecule that we produce, and this is what we're going to see. So we need to be able to set that stage for the building of the sugar molecule. So here's a little analogy. Have at it. Enjoy it. See if you can make some sense out of it. It actually kind of works pretty well. So a little fun fact. So even growing plants indoors, you're still using sunlight to do it. You're just using it, a version that was stored in the chemical bonds of fossil fuels that are now being used to produce electric lights. So just something to consider, that even the light that comes down on us was once sunlight at one point in time. Sort of, kind of. Cool to think about. So it's really important to think about these in the ways in which what goes in and what comes out. So if you can kind of keep that and kind of keep a flow chart going on that, it will really, really help, you know, kind of keep your head around these. So input versus output, what goes in, what comes out. Inputs necessary in the light-dependent reactions. Light, ADP, and phosphate, because we need to make AT. If we're making ATP, we need those two things to go in. We need NADP plus. Okay, source of that is going to come from the Calvin cycle, because we're going to produce that in the Calvin cycle. And water. Okay, what do we get out of it? ATP molecules, NADPH, oxygen. Boom, done. So we're going to end this podcast here. I decided to break the photosynthesis up into a two-part piece. One, to avoid mutiny from you guys. And two, I think it'll make things a little bit more clearer if we break up the light independent from Calvin cycle. So look for the Calvin cycle in the next one, as well as some sort of ways in which plants actually have adaptations for when CO2 is in low amounts, or if they live in really high temperature areas and some things like that. So again, I'm just going to split it up. So have a great night, and we will catch you guys later and talk through all of these processes. So take it easy. Bye, guys.